Oh, you started recording? Then you had to switch it off also. We just wait for 3 to 4 more minutes. Sadia, you can use the chat window to answer. Could you please take care of the lobby? Should we go ahead and get started, Sadia? How many people are still there? 20 are in, in the class, and okay, so I think let's go ahead and get started. Assalamu alaikum. Good evening, good afternoon to some of the countries where it's afternoon. Here it's evening rather than the night starts here. So it's 6:05 p.m. in the evening. Obviously, this is an online training course on NGS data analysis and coupled with some machine learning stuff. We made up this document. That's a guideline for us. So make sure everyone hears me clearly. You guys follow? So, do you guys hear me?
Yes, sir.
Yeah, okay.
Yes, sir.
Maybe we can have a brief introduction from you guys real quick. Your name, where are you from? Anything else you want to tell us about you? We'll be starting with our international friends. Who's out there? You can, if you have problem in listening, if I'm not audible or clear, you can switch on your mic and tell us, or you can post into the chat window. There is a hand button in MS Team. I guess some of you might be new with MS Teams. So that MS Teams is like Zoom. The difference between Zoom and MS Team is that here we can have, since it's our organization provides us in Zoom, like on the one which we are using, it does not go beyond 40 minutes. We need to purchase that, but this one is already, we have it licensed variant from our organization. From sites, thanks to them, we've been using in COVID days, and here we can have your test, your grading, your quizzes, assignments. I mean, not like a regular class, but maybe we can have some fun activities with each other. So it's kind of more interactive, like interactive and more um, good as far as the classroom teaching is concerned. So obviously this is also a classroom sort of a style teaching which we would follow. So here we have, right, so maybe we can have the introduction from where are international participants, Sadia, at the left. Ahmed Haik, maybe have your introduction. Ahmed, you joined us very first. So as soon as I started the meeting, Ahmed was there. Ahmed is commenting on the chat window. If your mic is working, you can comment on the chat if you want to, and maybe would like to know about you. If you can tell us about you, very welcome and thanks for joining us. Ahmed, are you there?
Hello, sir.
Yes, Gigi.
सॉरी सर वो असल में आई वांटेड टू चैट बट वो मेरी चैट विंडो वो रिफ्रेश हो रही है मैं बार बार उसे रिफ्रेश कर रही हूँ लेकिन वो पता नहीं हो पा रही है सो आई वाज अनएबल टू चैट सो
Okay.
आई थिंक आई शुड गो फर्स्ट
Yeah, please go ahead. Gigi.
Okay, Assalamu alaikum, sir. I am, and everyone also. I'm Lala Ilahi. I'm doing my MPhil in biochemistry. I've earlier joined your NGS workshop as well at Northwest uh, Institute Hospital जो था.
Very nice, very nice. You are from Peshawar?
Hello.
Gigi, you are from Peshawar? The one we did at Peshawar, right? Gigi, we can hear you.
सर असल में रीजॉइनिंग हो रही है ना आई डोंट नो व्हाई इंटरनेट कनेक्शन जो है इट्स वीक आई डोंट नो यहाँ पे तो सही है
Maybe on our side. My apologies for that. Yeah, please carry on. We can hear you.
ओके Okay. Actually, I'm doing my MPhil in biochemistry. My work is on an aging model, especially on animal model. So
Very nice.
I wanted to go further into the molecular biology side, especially in terms of next generation sequencing. And as we know that it's an AI era, so machine learning or Python, it's quite necessary when it comes to data analysis.
Yeah, we start with very basics and finally we end up with some of, not all, but some of the intro sort of ML analysis. But our focus will be mainly on NGS data, RNA sequencing, or the variant calling data. How can you apply classification? So that will be there. Do you guys hear me?
Yes, sir. पता नहीं sir, it's connection issue. It's really bad. वो बार बार
Is Is it on? Is it on my
आवाज कट
side or?
जाती है और
जी जी. The rest of you, is that? Am I laudable and clear? Or it's only with Lala? The rest
maybe
of
it's
you,
with
am I doing?
me. I don't
जी
know.
जी. बाकी
You
लोगों
are
को
audible.
आवाज ठीक आ रही
It's clear here, sir.
है? जी सर. Yeah, rest of you are hearing clearly, right?
Yes.
Yes, we are here.
Yeah, a bit low. I realize that. I tried to connect with the external, but maybe I can ask Sadia to give me the Bluetooth. So she has put it somewhere. I could not find it. I got another one, but that also has an issue. Uh, Till then, let me send her a message. I posted on the group, so she has two numbers. Maybe she can see that. Okay, so that's what it is. Who else wants to speak in the meanwhile till my mic is fixed? So rest of you can speak and chat with each other. So who else? We have Dr. Iqbal from ICCBS. Are you around, sir? जी Dr. Iqbal.
Assalamu alaikum, sir. I think I should introduce myself.
जी Dr. Iqbal, are you around? जी Hamad, would you like to say something?
Yes, sir. I wanna give my introduction.
Yes, please go ahead.
Yes, sir. Myself, Muhammad Ahmad, by BSA 3A student, and I just joined the machine learning course, and I am really excited to see how it connects with what we are studying in our class, and also I am looking forward to learning and working with you in this machine learning or like what we call databases.
Interesting. Very nice. Thanks for joining. He's our undergrad
Yes,
in
sir.
his sophomore years, second year.
Yes, sir.
Mm-hmm. जी Dr. Iqbal, are you around? Miss Naima, MN. जी मरियम नईम, आप बताएं.
अस्सलाम वालेकुम सर.
अस्सलाम.
ये सर मैंने अभी ज्वाइन की है.
जी जी बोलें.
सर वैसे ही माइक ऑन है. आप मेरा इंट्रोडक्शन चाह रहे हैं कि मैं अपना
जी जी
इंट्रोडक्शन दूं?
प्लीज गो अहेड.
जी सर, मैं पीएचडी कर रही हूँ कैंसर जेनेटिक्स एंड थेरेप्यूटिक्स में कायद-ए-आज़म यूनिवर्सिटी से. मेरा काम मॉलिक्यूलर
इंग्लिश में करें.
ओके सर.
We have internationals here
Myself
also.
is Mariam Naim, PhD scholar from Qaid-e-Azam University, and my research topic is molecular characterization of pancreatic cancer. So basically, what I am doing is the analysis of the exome sequencing and transcriptome sequencing. Plus, already I am doing the few types of proteomics using few types of proteomic tools as well. So I have a deep interest in next generation sequencing data analysis, so that's why I joined this course. Thank you.
जी thank you so much. And who else? Just switch on the speaker. Whoever is available. We have Myhob Lalmi. Did I pronounce it correct?
Yes, almost, sir.
Almost. So how do you pronounce it?
It's Mihub Lalmi.
Ihub. Very nice.
Yes.
What does that mean?
It's talented or gifted, something like that.
Oh, thank you
Yeah.
so much.
Hello, everyone. I am
Very
from
nice
Algeria,
gift
but
God for us.
currently,
Our international participant from Belgium. Where are you? Qatar?
Me?
Uh-huh.
I'm Algerian, but currently living and working in Qatar.
Very nice, very nice.
Yeah. I'm
Very nice
working
to
in
see you.
the preventive reference lab as a medical
Mhm.
laboratory technologist in the molecular and sequencing department. We implemented recently NGS in our lab. That's why I'm here trying to learn with you guys.
Thank you so much. Thank you so much.
Thank you.
I hope you will participate actively and learn. We will learn and gain knowledge from each other. And basically, this course is more skill oriented, right? So knowledge may be we have the theory lessons that in the beginning, but mostly would like to do the hands-on part where we'll be using all those tools, whatever we know. So we try to give you the skills on that, inshallah. And I
Inshallah.
think you are friend of Ahmed. Like there is another guy, Ibrahim. Most likely he connected you with us. I hope.
Yes,
Yes,
sir.
hello.
So there is another friend, I guess, Ibrahim. Is he joining?
Uh, I'll try to contact him.
So you too, I guess. He's in Belgium, right?
Ibrahim.
Ibrahim, do you know him?
Yes, I know him. He's with me here in Qatar.
Yeah, yeah. So both of you in Qatar, right?
Yeah, both. Yeah, we are working together actually.
Oh, nice. Thank you so much. Tell him that we are just starting, and there is another one, Ahmed. Where is Ahmed? Ahmed, hi. I
Yes,
think
hello everyone.
Ahmed. Yeah, would like to hear about
Can
you,
you hear
brother.
me?
Yes, please.
Can you hear me? Yeah.
Yeah.
Well, I'm Ahmed Haik. I work at the RSS, Royal Scientific Society, at the Biosafety and Biosecurity Center, Molecular Virology Division. We do research about viruses, detection of viruses. We do Sanger sequencing, and we're looking forward to implement NGS sequencing
Yeah.
also.
Very welcome, brother. You're from Jordan, right?
Yes, from Jordan.
Yeah. Nice to see you here.
Nice to see you
Okay.
too.
So who else? Yeah. We can go at random. Iqbal Qureshi Saab, are you around? Okay, Miss Falak Abasi. And then we have Mariam Naeem from our previous course. We have Aksanaz. Anybody who is around, I'm just calling the random names, whoever is displayed on my screen. And Mahanur is there. Yes, please. So I see some old students also. Who is this? Aisha Siddiqa. Okay, so maybe I can refer you to the chat window. Some people have the issues with the mics. Or maybe on our side, I have the issue with the sound listening. So I'm pretty sure. Yeah, Aisha. No, we were not able to hear you. Maybe you can write it. No issues. Maybe we can have that introduction. If you want to share your names and details, people can connect with each other. So we'll be together for like 5 to 6 weeks. Maybe we can extend that depending upon if you are learning and if you are more excited about it. Previously, we had a course on data analysis that started with very, very basic statistics. And then we went through different tools and finally we ended up with Python and ML stuff. So people were really, really excited and really wanted to go deep into the thing. So it took us like, I guess, more than 2 months. Initially, our target was to go for like 4 to 5 weeks, but we ended up maybe more than 10 weeks. So time is not a factor here. We've been together. inshallah, it's a connectivity, this networking, beginning of a long-term relationship with each other, inshallah. And we'll tell about the company or the organization which we are representing, what we are doing. Um, we are doing for different charitable causes. You are part of it. Thanks for joining. Learn NGS and also help to our community services projects. That's the message. That's just a brief online training course outline which we made. We want to develop an understanding of next generation sequencing data analysis. So this course is mainly about data analysis, right? Not about data generation. For that we need to work upon, okay, so for that we need to work upon on NGS sequencers. They are beyond our reach, they are expensive. We cannot manage them, we cannot run them. We don't have in our organization, but we do have with our collaborative organizations like ICCBS, etc. Maybe the people in Middle Eastern labs, they might have the sequencing facility over there. While I was doing my PhD at the University of Illinois, Champaign-Urbana in USA, we had a central sequencing facility, WMCAC Center, that served around 60-65 different institutes and organizations, and we had to wait in queue for the sequencing of our samples. So it took us like 3 to 4 months over there. That's how in those modern countries people are running those machines. More data you put, the cost goes low. Good old days while I was doing my PhD, the cost was around $1,000 to $1,500 a sample, and now it has been reduced to less than, I believe, 400 to 500 US dollars per sample, right? But again, it depends upon which machine you are using, how much data you are processing. The next objective is to train the fellows on developing skills in data analysis. So we'll be using Linux. Linux is basically an operating system, an environment. Most of you are familiar with Windows. Windows is MS Windows, Microsoft Windows. Linux is another operating system. Operating system is the thing that runs computers. Computers, when they are made, they are all hardwares, right? Circuits, hard drives, processors, RAM. These things are then connected together, and there is a software entity, and then becomes the softwares, the tools which we use on them, like Excel, like R and Python. These are all softwares. So there's an entity that connects hardware and software. Uh, Basically, it's a software itself made in a very low level computing language. We'll talk about the computing languages, or what is a low level language, what is a high level languages. Just give you a brief idea. Low level is computer understandable. It's hard for humans to understand and handle, but good programmers, they do that, and people used to do that in the past. Python and R is like very high level languages. It's just like you are talking to the computer, you are giving them instructions, you are typing some commands which are very easy and handy, and they are just like our language, the way I'm talking to you and you are listening. So that's called as the high level programming language. So OS was written, or they are uh, prepared on those very low level languages. These are the entities that connect the hardware with the software. So Linux is an operating system. Windows is an operating system. You guys are familiar with Mac. It's an operating system. In Apple machines, the machines are coming from which company? Apple, right? Microsoft is more familiar, so Windows is everywhere, but since our tools are mostly, they are running on Linux. Linux is a protected environment. Basically, your data is protected. It's good for data protection and security. Other thing is that Windows is something that is commercial. It's expensive. So Linux is an open source thing, so people, they can develop the solutions, they can develop the tools, and they can share with the communities, right? So before we move into R and Python, I intentionally put a couple of hours on Excel, Microsoft Excel. We do that data analysis to understand the dynamics of the data, and then once you learn doing data analysis in Excel, learning R and Python becomes easier for us, right? So we'll process the data in rows and columns, and then when we move to R, again we have to process the data in those two dimensions, rows and columns, same in Python. So since we switch on from Excel, we move on to R and Python, so it becomes easy for us to process that. And then we give you an insight of machine learning and deep learning. Both are type of artificial intelligence, but we call it as artificial intelligence, that is derived from the similar concepts of real intelligence which humans possess. And humans, they try to make something, some machine that was an effort that started somewhere 60, 70 years back. People started thinking of making a machine that should mimic the human actions. So computer is also like that. Or okay, it processes the thing, it takes the inputs, right? You give the input and then it processes that inputs, like you see the things from your eyes and that thing is then processed in your brain, and then you do some reactions, and then in computers you do, like computers, they give us the output. So there is an input and then there is then throughput, that is the processing, and then there is an output. So these three things are there. So and then now we got computers which are developed that are intelligent, so they can think, they can process, they can do decisions, right? So that thing is covered in this third part. But before we go into that, we will understand a basic understanding of this sequencing. We start with DNA, and then we talk about sequencing, and then we talk about the next generation sequencing, right, in detail. So these are our objectives, and we set our objectives on weekly basis. Course overview and policies, that's just an overview I'm giving you, and there is no such written policy. It's an interactive course. You can interact, you can make it a discussion forum. We can learn from each other, right? Some people on other parts of the world may be doing something which are very advanced, right? So maybe you can share with us. So in the end, maybe we can have some interesting projects which you guys can work in groups and demonstrate. So in this way, we will have a mutual learning from each other also, right? So introduction to NGS will start in a moment on my PowerPoint slides. So slides will be shared, books will be shared, video tutorials will be given. For the hands-on sessions, setting up the tools, we might have additional sessions by Sadia. She's our lecturer as well as she's my PhD student, very talented, and she and along with the other students, they're the ones who deal with these tools most part of their time here. So I'm a professor, so I got other assignments to do, and maybe my knowledge or my skills may be a little bit old, but my experience and I believe the communication skills which required with the passage of time by the grace of God, that covers that. But obviously some of the tools are really new and emerging, like you people might have heard about Docker. So she's interested to cover that part also, and yeah, I have no issues if even if it goes beyond our regular schedule, as long as you guys are enjoying or you guys are staying, we will do that. The hands-on activity during this week, obviously this week means Monday and Tuesday, these are my sessions, my timing 6 to 8, right? And then you can have extra lab session with Sadia also. Maybe we might need one before we go into week 2, because for week 2 we probably need Linux. So we need to set up Linux on your machines, on your laptops, right? So yeah, I think some of you might have accidentally switched on your mic. Please switch it off. Okay, so in the second week, so these are the some resources which are there on which we can have this data set. One of them is Gene Expression Omnibus, Sequence Read Archive, where you can have their short read data. Even the long read data is also coming, so Sequence Read Archive is there. Gene Expression Omnibus actually started for microarray dataset, the gene expression, where we talk about the RNA data. Actually, DNA makes RNA and RNA makes protein. So when you talk about the gene expression, it's actually the protein. But here in this course, we are not going into the proteomic domain. So we are mainly focusing on genomic as well as transcriptomic domain, that is the RNA. And then people realize, like, if you study RNA, that will be okay and that will be good, because studying RNA was comparatively easier as compared to the proteins and cheaper, right? The techniques like microarray, and later on when NGS came out, it was not replaced, but actually RNA sequencing has taken up a pace over microarray. So initially that database was there for microarray datasets, so gene expression data mainly from microarray. Over there they used to capture the messenger RNA. So mostly these were the messenger RNA datasets. Other RNAs are also captured, right? But mostly these are messenger RNA that are involved in gene expression. So when you say gene expression, that means transcription and translation. So GEO, Gene Expression Omnibus database of NCBI, is where we can get a large amount of data. Then we have the raw data, that is the short read data coming from those NGS sequencers. We call them as short reads. Read is actually a fragment when it comes out from sequencer, and a sequenced fragment is called as read. So there is a repository in NCBI, SRA, Short Read Archive, where you can have that data, the raw data, the sequencing data, mainly from NGS sequencers. And then we have the organism-specific databases. So we need the gene models for RNA-seq studies or for our variant calling studies in our projects. So for there we can go to specific individuals. So we have human genome on UCSC, University of California, Santa Cruz, their database, and we have TAIR, TAIR or TAIR, that is for Arabidopsis thaliana, right? A model plant. Similarly, we have separate databases for yeast and all other famous model organisms. We would just like to see these databases also during this week. In week 2, when we have the introduction and we know more about that NGS technologies, we will be then started doing the data analysis. For that, I would prefer going straight away to Linux, but my core trainer believes that maybe we can give them an online resource. One of them is called as Galaxy. So that is not recommended for your actual analysis in real life, but for the beginners who are starting the analysis, I think that is good. Even if you have the small genomes, maybe you can process them like mitochondria or chloroplast or viruses or bacteria. But still, some people argue that we should not use that for our real analysis. For the real analysis, we should start Linux and R and Python as early as possible, right? So I hope by the end of this course, you'll be doing that, okay? And then we talk about the variant calling, sequencing and resequencing. When you are sequencing something for the first time, that is called as sequencing or de novo sequencing. Over there, you need to do what we call it as assembly, right? While you do sequencing, you actually disassemble your original genome or whatever that target DNA is, maybe a part of a genome, maybe a chromosome, maybe the exons. When they are put together, we call them as exomes, right? So we actually disassemble them. and then we sequence them. So we break them into short fragments and we sequence them because we cannot sequence that whole big chunk in all together. So that's why we need to break it into short fragments, chunks. Okay. We'll cover that in PPT slide, but this is just an overview I'm giving you. So don't worry, we will see these things again and again and again. If you are hearing it for the first time, you might be having some difficulty, or might be, don't be scared with these terms, right? Uh, yeah, with the passage of time, you'll be familiar with them. It's like you are learning a new language, a new dictionary. So I'm adding more terminologies to that. So that is the language which we have. My professor used to call it lingo. There is much lingo here, right? And annotation is when you assemble a genome or a transcriptome or maybe a chromosome, then you want to know what it contains inside. So that's annotations, right? Like you get a text from some old language, from some history, right? Latin or some language that is pretty old, and it has maybe the scripts, or maybe something like holy scripts in Hebrew, and some other language. um, Arabic is still valid, right, and alive, but some of the languages are gone old. So what people used to do is they get those scripts and then they try to translate that and interpret the actual message from them. So we believe that DNA is also a language, right, that has alphabets in it, those 80 GCs, right? Amino acids are also making up a language, so there are more alphabets in that, right? So proteins are also, sort of a language. So when you sequence them, when you assemble them, then what message is written, right? Where you can find the start of a file, and where you can find the end of some script, and the body of the script, right? And the sentences and the stanzas and the passages, right? So all those things are called as annotations. In terms of genes, or in terms of DNA, finding genes, finding coding regions, non-coding regions, right? Intergenic regions, introns, exons, all those things are called as annotations. Okay. So assembly and annotation are part of this genome assembly project. Next one is, while we do variant calling, variant, as the name indicate, that is coming from variation, right? So variant is a variant of something, okay, an alternative form of something. So there is a sequence and there is another sequence that is pretty close to it, but it has some variation in it. So that thing is called as variant calling, right? So different variants of a sequence, okay, some gene is not normal in an individual, so that means he has some variant that is deficient, or that is having some abnormalities in it. So obviously we need to find those variants, so that variant calling is also done here, right? And mostly we do it on exome data. When we say exome, that comprises of coding sequences, exons within those genes. So that part will cover here. And then within those parts, we will also apply ML on variant calling data, right? Then next we move on to transcriptome sequencing. In the hands-on activity, obviously we will go with genome assembly and variant calling tools, and there are a lot of them. We select few of them, and depending upon which species you are working, you would look for whichever is recommended. You will go through literature and then you will use them, but we'll try to use the ones which are the most applicable to the most of the scenarios, right? The generic ones. Transcriptome sequencing is obviously, as the name indicates, the genome makes transcriptome, DNA makes RNA, RNA is transcript, all transcripts put together makes a transcriptome. So that technique got famous as RNA seq. This word seq is actually representing the sequencing, and sequencing not by ordinary or classical methods, but sequencing by something like NGS machines. Okay, so that's why this word seq is there. You will see exome dash seq, right? Metagenome seq, right? RNA seq. So that seq word indicates that This thing is coming from sequencers, which are modern day sequencers, NGS sequencers. So in this RNA-seq analysis, we will spend more time on it. We'll talk about the differential gene expression testing. A gene has different expression in different individuals, in different conditions, right? Patients versus normal controls. What is the difference here? Which gene is going up, which gene is going down? So that thing is called as differential gene expression, DGE, right? And after you narrow down to a group of genes which are DGEs, differential or DEGs, we call them as DEGs sometimes, differentially expressed genes, right? Differential gene expression is the method, right? So DGE testing. The genes which you get in the end are DEGs, differentially expressed genes, right? So you take those DEGs, right? And then you get their gene ontology annotations. So most of the genes are performing which function. So those functions are again, we are getting a message here, right? If it's a cancer patient, so we see that most of the oncogenes are upregulated. Their expression is higher. Comparable to that, maybe most of the tumor suppressor genes are downregulated. Their expression is reduced, right? So that is the differential gene expression. So since gene ontology annotations, they are telling us that the overall expression is going towards either cancer progression or cancer, control. So all those things are then covered here in those gene ontology annotations, GOAs. So we study that part also. Gene networks is another interesting and one of my favorite areas where we put the genes together and we take their gene expression counts, their values, and see how do they interact with each other. Which group of the genes are positively correlated with one another, which amongst them is negatively correlated with one another. And in this way, maybe we can try to infer the cause and effect. We call it as causality within those networks, right? So we believe like the genes are activators to each other, or some genes are repressors to each other, right? So negative correlation is repression. Positive correlation is an activation, right? So that part is then covered here, gene networks. And again, we'll apply ML and RNA-seq, one of our studies done by our graduate students. So that's what we would like to highlight. In the hands-on, obviously, we'll use R and Python and whatever. If time permits and people are interested, and I believe that some people are interested, highly interested in metagenomics. Metagenomics is like you can take, a group of genomes, right? Not intentionally, you take everything and there are the DNA in it, there are multiple genomes in it. So that's why that term comes out, metagenomics. You take, for example, the samples uh, from some individual, like you can take the samples from his gut, or you can take the samples from his blood or any other body parts, and you observe there is a group or cluster of bacteria over there, right? There are different types of bacteria that are there in our gut, so many of them, right? And some people believe, the people in metagenomics, they believe that we are more bacteria than humans. Why? Because the amount of DNA possessed by those bacteria in our gut, that is way more than the DNA which we possess in our normal body cells, right? So some people believe that those bacteria, they drive us, they drive our 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 living patterns, right? They determine our moods. You get good diet, bacteria is happy, you are happy, right? So those sort of things. Anyways, very interesting area, and I hope we will have enough time to cover that. But again, I would say I'm the one who is not restricted with the time, so if we go beyond our normal time, so please bear with me or permit me, right? So we have lot many things to cover, lot much knowledge to share. So don't worry, we'll give you the good product, right? So let me start with the lecture part now. We start with the slides. These slides will be shared with you. Maybe we should have shared before the class, but then probably you will be more looking into or going through these slides by yourself. Let me switch that part. So can swap the display here. This one. All right, so that's our first lecture about ComSets. You guys must be wondering what is ComSets. ComSets is Commission on Science and Technology. This is me, of course. I think I skipped my first slide. So Dr. Wasim Heather. American style is we don't go with doctor or sir or Hazur, Kipla, Motram. It's just a PhD degree, right? So that's me, Wasim Heather. An associate professor at Computational Biology and Bioinformatics Group. We call it as CBBG. CBBG stands for Computational Biology and Bioinformatics Group at the Department of Biosciences, and this is ComSets University, Islamabad, right? We also came up with a humble initiative. We call it as Next Gen Solutions. The word NGS is also here. We kept it intentionally for two reasons. NGS also stands for Next Generation Sequencing, and it's also Next Gen Solutions. The problems to the solution to the problems of our next generations, right? That is the poverty creation, and that we can do directly or indirectly, giving them skills, make them able to apply those skills, and on good doctoral and scientific positions, number one, and also directly by by helping some of the underprivileged kids in their education. So that's what we are doing. So that's why we call it as Next Gen Solutions, the name of our group. About ComScience, the Commission on Science and Technology for Sustainable Development in the South. Uh, that was a vision from one of our Nobel laureates, from I guess the only one in science, by the way, from Pakistan, Professor Abdul Salam. So his vision was like there should be an organization representing mostly the developing nations, the poor nations, which are not developed, not in terms of money, but scientifically and technologically. And now it got 27 member countries, mostly the Muslim countries, but some of them are coming from, since most of those countries are coming from the southern hemisphere, so that's why it's called as the Commission on Science and Technology for Sustainable Development of the South, right? Maybe some people might be familiar with ComStech. ComStech is an organization, again an international organization that has more countries in it. So that has, it is chartered through OIC, Organization of Islamic Countries. So it has a standing committee on science and technology. So ComStech is different, ComSats is different, right? But they are like sister organizations. And I think there is in Algeria, somebody was from Algeria also. So he also has, I guess, ICISCO, one of them. There is another third sister organization. So I think that is similar to, a little bit smaller than ComSat. So there is the ComSat, and then there is ComStech, and there is ICISCO, right? So these are the organizations. So mostly the underdeveloped countries, mostly the Muslim countries in it. So that's our organization which we are representing. This organization came up with a Center of Excellence in IT, Information Technology. That's where ComSat's Institute of Information Technology started in 1998. And this one came in 1994. The commission materialized. Obviously, Professor Saab had this vision and had initiated this somewhere in 70s, 1970s. But it got established in 1994. Like some of the countries, they sit together, and then they decided to start this, and headquarters is there in Pakistan, right? But most of, I think some of the fellows are connected with us for this course through our friends and brothers who had this joint ComSets training. We had the people from different ComSets countries. Right, so that's our team. All are foreign qualified, like, and then half of the people are working in genomics and half of them are working in proteomics. In bioinformatics, the major two divisions are genomics people and proteomics people. On the left, these are the proteomics guys, except for Han. On the right, we are all genomic people. I guess Zoya and Farhan, they may be switched. So these three and this one, four of us on genomics and transcriptomics side, and these three and this young lady, Zoya, these four, they belong to proteomics. The next gen solution is just an effort, a humble effort to do those trainings and do some community services projects. Just a glance of our trainings, which we did, and yeah, right. And we trained like more than 1500 and some of the community services projects where we help the rural kids as far as their education is concerned. We do medical camps, sometimes we do food bag distributions. We supplied computers to few schools for those underprivileged children. We also came up with vocational training centers where we gave training to the girls regarding this tailoring and stitching. Let's get back to the work. So about DNA, everyone knows that there is this darn thing and that's called as chromosomes. What are chromosomes? The bundles or the packets which are visible at the time of cell division. Under normal routine, this DNA lies in nucleus, not that visible like those bundles or packets. You only need to uh, you know put the things in packets or bundles when you need to divide it. So at the time of cell division, that whole genome appears in the shape of those bodies we call them as chromosomes, right? Everyone knows that chromosome means colored bodies. So somebody who identified them very first time, he had a eureka moment and he said chromosomes, right? These color bodies, okay, because they get some colors under his microscopes. Probably he was using staining techniques, so they were stained. So anyways, you zoom in till you see there is a thread like thing and that is wrapped on those bead like things. So the beads and thread, beads are what we call them as, these are made up of proteins, histones, and the thread is, then you zoom in till you see that then it becomes two threads which are wrapped around each other like two snakes wrapped around each other, and in between them we see there is something like ladders. Okay, so there is a ladder and there are those threads, green and red. By the way, this is a cartoon picture, right? That is just to give our concept. One bad thing about this is if somebody is colorblind, green and red might not be recognized, but I adopted it from this guy, so it's his fault, not my fault. So till you zoom in, till you see that you have those alphabets. They are not these alphabets written like this. That is again, as I said, this is a cartoon picture, and that's what we have for our perception, right? So those alphabets are actually coming from what we call them as nitrogenous bases. So DNA or deoxyribose nucleic acid is built upon these molecules. The generic term is nucleic acid. One of them is ribose nucleic acids or RNA, and one of them is deoxyribose nucleic acid, the DNA. Right? So both are built upon these 5 carbon sugar molecule called as ribose. So ribose on the right, deoxyribose on the left. So what we see in ribose is there are 5 carbons. So this is carbon number 1, this is carbon number 2, this is carbon number 3, 4, and 5. These carbons are numbered, right? 1, 2, 3, 4, 5. You might realize that there is a dash like thing written here. We call it as prime, 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. Why we needed to do that? Because we had this numbering, and I think some of you might know, we had that numbering on those nitrogenous bases. They are numbered as 1, 2, 3, 4, 5, 6, 7, 8, 9. right? 1, 2, 3, 4, 5, 6, something like that. So when you put them together, you have number 1 here, but here you put 1 dash, so 1 prime, right? So these are numbered 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. Each carbon position here is important. Why 1 prime is important? Because when it makes DNA or RNA, before it makes that, it comes up in a shape of what we call them as nucleotides. So those nucleotides, they carry three things, that 5 carbon sugar molecule, ribose or deoxyribose. That is the main core thing on which this structure is built. And there is a nitrogenous base that comes and attaches here at position number 1, right? So position number 1 is important because nitrogenous base attaches here. Why position number 2 is important? Because here on ribose it's OH, here it's H, right? So in deoxyribose, that O is gone. It is deoxygenated, right? Or in other words, let me say it is suffocated. So this one is suffocated, this one is not suffocated, okay? So here you have O, here you do not have O. So that is the difference between ribose and deoxyribose. At position number 3, why it is important? Because when those nucleotides are added in DNA or RNA, that next nucleotide comes in and attaches here. So that end is also called as 3 prime. So 3 prime end, we call it as 3 prime end, where the new nucleotide attaches here as well as here, both in DNA and RNA. 4 is important because it makes that oxygen linkage, right? There is an oxygen molecule here, so 4 and 1 are linked together with the help of that oxygen things, and that gives this ring a 5-cornered shape, because that 5th carbon is not here in the main ring, that is outside, okay? So that 5th position is also important because the phosphate molecule comes in and attaches here. So each carbon position here is important. Number 1, because anybody, you can type it into your chat window also. Maybe I can give you a break and you can think on it, right? And then we can come back and resume, right? So just tell me why position number 1 is important. I can open up the chat window and see what's happening there. Yeah, so position number 1 is important, or maybe you can switch on your speakers and just speak, just to make you active and alert. You follow?
Because nucleotide is attached to carbon number 1 first carbon.
Yeah. And I see a question. Can you share the basic statistical manner? Yeah, when we start talking about the data analysis, we will cover the basic statistics. Don't worry. Right? So we try to put in the ingredients that may be necessary for some of you, somewhere for most of you. But like some people might be good with molecular biology, but what if a computer scientist is sitting, if molecular biologists or biochemists are feeling bored of it, maybe you can review that part, this first part. I mean, this chemistry is ugly, but for somebody who's coming from CS, or sometimes people join from electrical engineering background or computer engineering background. So anyways, for them this information is important. So we'll have those ingredients, don't worry. What next? Yeah, nitrogenous base attaches, and Aisha Siddiqa. Thank you so much. Yes, very right. Position number 2, OH here and H here, right? And that is actually a distinction between ribose as well as deoxyribose. So that makes this thing that contains this one as RNA, and the one that contains this part, the suffocated variant, that is DNA. So DNA is suffocated. Ribose is, or RNA is not suffocated. What if you took the second oxygen from here? And that's important because it's coming next when we talk about the DNA sequencing or Sanger sequencing, that thing will be covered. So if you take the second oxygen, then it becomes dideoxy, DDR, dideoxy ribonucleotide, right? So dideoxy. So maybe I can just give me 5 minutes. I can hear one azan, and then you can take also a break and you can get some fresh air. We'll see each other. So azans are going on. So Hello, are you there?
Yes, sir.
Okay, so where were we? जी हम क्या कर रहे थे?
सर
Where were
आपने
we?
डाइडिऑक्सी राइबोस के बारे में बता रहे थे.
Okay, so we were talking about carbon 1, nitrogenous base attaches here, 2 is where it's oxygenated and deoxygenated, 3 is the one that, and then that OH is also important at 3. If you remove it, this O from here, then it becomes dideoxy, then next nucleotide does not attach into it, and then there is number 4, that is where it is um, important for this oxygen linkage, and this 5 carbon, this 5 cornered, I'm sorry, ring is formed, and 5 is important because phosphate group attaches here, right? So yeah, in DNA, these nucleotides, adenine, guanine, cytosine, and thymine, they are there in DNA. The nitrogenous bases, there are 4 of them: adenine, guanine, thymine, and cytosine. And then there is the 5th one, that is where this thymine is replaced by uracil in case of RNA, right? So that is there in RNA. In DNA, it's thymine. So cytosine and thymine are single ringed, whereas adenine and guanine are double ringed, right? So, and then these nucleotides are formed where this nitrogenous base is then attached to this sugar molecule, and then it becomes nucleoside. So we have the nitrogenous base as well as the sugar molecule put together. The thing is called as nucleoside. So adenine is a nitrogenous base, and in nucleoside form, this thing becomes adenosine. Here, just remember one thing, that which thing is causing this name here, which thing is causing this variation here, because this sugar molecule is the same throughout for all those nitrogenous bases, but that nitrogenous base is different. Phosphate molecules are also the same, right? So those nitrogenous bases, when they are added to those sugar molecules, they give them their names, or they give them their distinctions, and that variation or variability is coming from them, right? So adenine then becomes adenosine here. When you say adenine, that means the nitrogenous base. When you say adenosine, that means you have this both the things together, the nitrogenous base as well as the sugar molecule. So that thing becomes adenosine, right? So this adenosine then adopts a phosphate molecule. Generally it does not adopt one, but it takes 3 of them together. Those phosphates, they come in a triphosphate shape. So then it becomes this, since it's deoxy, so it's 2 prime, it's 2 prime position, it's deoxy, so it's called as 2 prime deoxycytidine. So now we call it as cytidine um, triphosphate, right? So here when DCTP, so here when it has one phosphate molecule, the way it gets incorporated into the DNA and RNA. So over there it's called as AMP, adenosine 5 prime monophosphate, right? So adenosine 1 phosphate makes it as adenosine monophosphate, AMP, right? So then these 4 of them, um, they are the ones, those that adenosine or cytosine or guanine. Okay, no issues, no issues. They give, it's okay. थोड़ा सा ना माइक की आवाज़ लो जा रही है, ये वाला भी चार्ज नहीं था. आप ये वाला ले लेना, इसमें इट्स ओके, बस साथ गुजारा हो गया. पहले मैं गाना चलो, वो मैं ये इस पे आवाज़ क्यों नहीं आ रही उनकी, ऑडियंस की आवाज़ इस पे नहीं आ रही, इस वाले स्पीकर वाले पे. Don't know. यहाँ से कुछ सेटिंग का आई थिंक इशू. ठीक है, आप जाएं, फिनिश योर क्लास. So we have those, this Watson and Crick, they gave us the model, the structure for the DNA in 1953. So according to them, the DNA runs in anti-parallel fashion. That 5 prime end is here. This 5 prime phosphate molecule is exposed. So one of the strand goes upside down, the other one comes in upward direction. So that's why they are called as, they are arranged in anti-parallel fashion, such that if you look into this strand, so that is the 5 prime end, or 5 prime phosphate is visible here, so that gives its end as 5 prime end. So DNA runs from 5 prime to 3 prime, right? So we read them from 5 to 3. So that gives us a directionality, because when this DNA strand is synthesized, it is synthesized from 5 prime to 3 prime end. So here is the phosphate thing, then there is sugar, phosphate, sugar, phosphate, sugar. That is what we call it as the backbone, the 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 threads, right? Are the bamboos, are the sticks within that ladder sort of a thing. And this thing is on the other opposite strand, it goes like 5 3 5 3 5 3. In the end, this 3 prime end is free. So this is 5 end, this is 3 end, 5 prime end, 3 prime end. On this side, 5 prime end and 3 prime end, right? So A is attached with those two strands are then attached with each other, so that anti-parallel movement gives them an opportunity for those opposite nucleotides to be attached in what we call it as canonical fashion. What is canonical way? A attaches with T, C attaches with G, A goes with T, G goes with C, and so on. So whenever these attachments are there, we call them as canonical pairing, or the normal pairing. And then we also realize that between G and C there is these triple hydrogen bonds, so they are stronger connectivity. And then A and T are having double bonds. So C goes with G, A goes with T. That means if you have one side of the strand, you can predict the other side. So that capability was utilized in one of the processes called as sequencing. And before sequencing, the DNA replication was understood. So what people understood, that during the replication process, the DNA unwinds, and DNA like it gets separated. Those two strands are separated from each other. So one of the strand that runs from 3 to 5 is read as a template, is a template. And then the opposite to that strand, the strand which is missing, that is the new strand, is synthesized. So DNA replication is actually a process where the old strand uh, is replaces its new strands with the free nucleotides which are present in the medium, in the cells, right? And you have the other machinery also. You need to have an enzyme called as DNA polymerase. We also learned that the DNA synthesis cannot start by itself. It cannot start with what we call it as de novo. So you need to have something that is already there. We call it as primer. So that primer is where it is a specific run of nucleotides. Normally we prepare those primers and then we attach to that DNA strand. They also gives us the specificity. So the place where we want to do sequencing, we design the primers against them. And then, for example, it starts from here. So the first nucleotide on the template was T, and it is replaced by A here. So G goes with C, T goes with A, C goes with G, and so on and so on. So whatever you have on the opposite strand, since you know about the complementarity, you know about the canonical pairing, the opposite side will be having that nucleotide that will be complementary to what we have in the template. So what you need primer, what you need free nucleotides, what you need DNA polymerase, and obviously you need that template. This template is run from 3 to 5 prime end like this, whereas the new strand is synthesized from 5 to 3. So that's why this directionality is important. But remember, as we covered before, that this nucleotide, the terminal nucleotide, where you are incorporating the new one, for example, you are incorporating this new nucleotide here. After G, you have to incorporate a C. So you have to incorporate a C after having this G here, and this G is, for example, we zoomed in and we are seeing that G here. So that G has this situation that it has at 3 prime end OH, uh, O is already present here. Then the new nucleotides, obviously in this case it was supposedly a C, it comes in a triphosphate shape, and those two phosphates, they are then cleaved, they are moved, and then this single phosphate shape, it comes and attaches here. So on one side those two phosphates are going away, so one of the sequencing technology is will use those two phosphates. And obviously somewhere on the other side there is a hydrogen ion that goes out, right? So that's hydrogen ion on one side is removed, and on one side that those two phosphates are removed. Remember, these two technologies will be used in in the future technologies. These two molecules, hydrogen ion and those two phosphate molecules, what we call them as pyrophosphate molecules, they are used uh, in two of the sequencing methods. One of them is similar to what we will see in the next slide, similar to Sanger. If you remove that O from here and it becomes a dideoxynucleotide here, dideoxy DDR, so then it becomes DDR, dideoxyribonucleotide, or terminal nucleotide or terminator nucleotide. So here you cannot add the new nucleotide. So that quality, and when those DDRs they were synthesized, they were used by Mr. Sanger, the Fred Sanger. So one of the famous sequencing method, and later on it got people came to know that, I mean, at that time it was just DNA sequencing, but since we got NGS, we used to call the previously present method as the previous generation sequencing, next versus previous. So that was sort of rude of saying, and the work of some big scientists like Fred Sanger as the previous generation. So people use the term then first generation. So that's the actually Sanger method, where what we used to do is we have that template, we have that primer, and we set up 4 reactions here. So in one of the reaction tube or mixture, we have everything normal, except we are using a combination for ATP. So we have a normal ATP plus dideoxy ATP, and rest of the nucleotides are normal. In the second one, we have everything normal, but a combination for CTP, normal CTP plus dideoxy CTP. And then in another tube, we have GTP, right? So we have GTP, a normal triphosphate, guanosine triphosphate plus dideoxy GTP, dideoxy guanosine triphosphate, right? And then we have in the fourth tube, we have TTP, normal plus dideoxy 1, okay? So we have combinations, combos, a combo for ATP here, a combo for CTP here, a combo for GTP here, a combo for TTP here. So what's going to happen here is when this picks up the first nucleotide, that should be C against a G, right? So if it picks up a normal C, that's fine. So chain will continue. But if it picks up this one, and obviously these are put in bulk, so there is a equal probability of picking a normal CTP versus a dideoxy CTP, right? So obviously we will have different reactions. So in the end, we will have those differential fragments here. So here the first nucleotide that was incorporated was a dideoxy CTP. So that's why the chain stops here. So we got a fragment of length 3. Obviously this primer we are not considering. So maybe we can say fragments of length 1. Then in this one, that is the fragment of length 2. Another fragment of length 5. So we have 3 differential fragments here. Similarly, in A tube, we have 2 differential fragments. In G tube, we have 2. Obviously, we will also have the full length fragments, but we are not considering them. So we will have the full length fragments also, the third type. They are in bulk, so we are not considering them, right? Even when we pour them here for this gel electrophoresis thing, they will stay at the back, they will not run this race. Then we have those 2 types of differential fragments in G tube and in T tube or T mixture. This is A tube, C tube, or mixture, or G mixture, or T mixture, or tube we call it, right? Or reaction. So A reaction, C reaction, G and T. So in T reaction, we have 3 types of fragments here, right? So when we have those differential fragments, we can separate them in a technique called as gel electrophoresis. So this is a gel here. We set up the gel, we connect them with the different terminals of the battery. Everyone knows that, people are coming from molecular biology. And finally, you separate them and they run a race, and there is an electrical gradient. By the way, it is not a this vertical gel. Normally it is placed in a horizontal plane, so this picture is vertical, so that's what we are seeing them. So normally we load those fragments here, and this way they run. So this is the smartest or lightest fragment. Obviously it is this guy. The technique is you just need to number them. So this is your number one, that's your finish line. So they are finishing here. So this is number one, this is 2, 3, 4, 5, 6, 7, 8, 9, and 10. So 10 bases we are sequencing here, and we have prepared 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So we have 10 differential fragments. So as many bases, as many differential fragments are needed. Okay. Let's suppose if your fragment, or if your total length of your DNA is 20, so you need 20 fragments, right? If you have 100, you need 100 fragments, 100 differential fragments. That's a lot. If you have 1000, you need a length of, for a length of a DNA of length 1000, you need 1000 differential fragments. So obviously those gels, they have a limitation, they have a restriction. Then next is, we know from which lane or from which reaction they are coming. So everybody you poured here was coming from this tube. So obviously these are all C's here. So this guy is C. This guy is, you look up, what is that? C. What do you have in here? Anybody? That's a, look here. That's an A. What about this guy? So that's a T here. What about this guy? Anybody? You can type your answer into the chat window.
Yes,
Right?
sir.
So this guy is a, right? Look up. C. Shabash. This guy is a G. This guy is C and C and G. And this one is A. So you nailed it. So here we are seeing them in black and white screen, and we are getting those different fragments, and they are there on that gel. And obviously gels were expensive, gels was tricky. They were also having, the later on we realized that they are having those environmental hazards also, some of the gels. So people try to get some replacement, and obviously we need to inspect them visually through our eyes. So people try to take their pictures of those gels and started putting them or linking them with some electrical system, with some computers, till Mr. Laurie Hood at Caltech, California Institute of Technology, he came up and automated this thing, and that's called as automated Sanger sequencing. And later on, when NGS came out, this thing then got renamed as first generation of sequencing, or first gen sequencing. That Sanger sequencing. Sanger was the method that was famous. Before that, there was also another method. Most of you might be familiar with that. So um, the Maxim and Gilbert. By the way, they were also students of Professor Abdul Salam Saab. So Maxim and Gilbert method was also good, but it has a little bit complicated method. So that's why people were a little bit, scared of it, or hard to apply that chemical degradation method. So this method got more famous because that was easy to understand and easy to implement. And then this thing was automated by Professor Larry Hood into what we call it as automated Sanger sequencers, or first generation sequencers. So now normally some of the labs, even our lab here, one of the labs, genomics, they have that automated Sanger sequencer or first generation sequencer. At that time, it was just called as DNA sequencers. Nobody knew that it was the first generation or second or third, because that was the only generation that lived there, that helped us finish our human genome project. But it was costly, it was expensive, and then till people came up with those NGS things and make it very cheap and very quick and very efficient, right? So what's happening over here is Professor Larry Hood replaced the gels with capillaries, and he replaced that black and white thing with the colors. So these dideoxyribonucleotides were given some fluorescent colors, right? So some chemicals were added to those nucleotides, and they were then labeled nucleotides. These were non-labeled black and white, and these were labeled nucleotides. Okay. So this one is, for example, I mean, green, or boys, we are colorblind, so we will just recognize 7 basic colors. So for me, it's green, and it's blue, and it's what's that, yellow, and this one is something like red or pink. But girls, they can distinguish them as, parrot green and, sky blue or pink or whatever. And there is an evolutionary reason behind that, right? Our dresses are also not that colorful. They are also either, yeah, we are black and white, by the way. Anyways, so here, um, one thing is happening. You are not loading them on the gels, and you are connecting them. You are taking all those fragments in just one cube and connect it with a capillary on it. And when liquids, they see a capillary, they start rising up. This thing is called as capillary action. Capillary action. So when they move up to the capillary, they go through those canals, they go through those tubes until they reach here. So when they reach here, the first fragment is the smallest one. So here is a detector in the camera, that is their movement, negative to positive terminal. Somewhere here there is a detector that's detecting, and they are coming in an order, in a sequence. So blue color first, that means it's a T here, C. This red color comes in, pink color comes in, it's a C here. What's that? Golden or yellow, it's a G here, again a G here, and an A here, and so on. So this way you sequence them. That was called as your Sanger sequencers or automated Sanger sequencers. Nobody called them as first generation till we saw this thing, what we call it as NGS or next generation sequencing. NGS, next generation sequencing, is a catch-all term used to describe a number of modern sequencing platforms. What are those platforms? What are those machines? Illumina, one of the company, Illumina came up with reversible terminators. So it was using that terminator things, but terminators were reversible. Remember here that dideoxy terminator, once you put it, you cannot take it out and you cannot continue the chain. But here they did not suffocate it. Rather than that, they did another trick. Rather than taking oxygen, they replace hydrogen with nitrogen, right? And then this nucleotide is added, the color signal is detected, and then you put water, and then again that H comes back, N goes away, right? And this thing is then it continues. So that's why this thing is called as reversible terminators. We will see them in a moment, the actual molecules. Roche, another company that used to provide you the chemicals and reagents, that came up with another method called as pyrosequencing. Pyrosequencing is where you are using a pyrophosphate molecule. What was a pyrophosphate molecule? Remember, when nucleotide is added, two phosphates, they actually run off from that place. So that thing is called as pyrophosphate. Okay, pyrophosphate. So those two phosphates, they run away. So that's why this thing is called as and you capture them, those two phosphates, that we covered that part, and I asked you to remember these two things, and I told you that there are the companies that are using them. So one of them was using the terminator like dideoxy, but it was not dideoxy, it was deoxy, but it was a reversible terminator, and that was used by Illumina. So these two phosphates, they go away and then they carry energy, and those two phosphates are then allowed to react with some other chemicals, and that produces illuminescence. What do you call it? The light signal. Okay, so that light is coming out from the power which we had in those two phosphates, and this hydrogen ion that comes out is captured by another company called as Ion Torrent. Third one is Ion Torrent. Thermo Fisher Scientific, that was the company that was using that. It's also called as ion semiconductor sequencing. Applied Biosystem, ABI, comes up with solid sequencing, though it's a history lesson. Mostly their machines are gone, they are obsolete. The name is uglier, similar to their algorithm was also tough to understand and implement. So solid stands for sequencing by oligonucleotide insertion and detection. I did not put that abbreviation down there because that's not important now. It's become obsolete. It's gone. Next generation sequencing includes these technologies. Okay, then we have the third generation. So since this previous one, when you call something as next, why we call it as next generation? Because that was a catchy term. When this millennium changes from the 20th century to 21st century, right, year 2000 onwards, so that term was catchy and it was there in you know marketing and selling different products. So next generation refrigerators. next generation televisions, right? So we had the televisions that was quite big and elongated, and it has a very, very ugly sort of a body. But now we got these smart televisions, LCDs and then LEDs. Nowadays, people are not that familiar with those televisions which we have seen in the previous millennium, right? So that NGS, next generation term, is because of that word became famous when this new era started. Okay, so that's why NGS term was adopted. So when you call something as next generation, what do you call the previous? The previous generation? Obviously, nobody dared to call it because Sanger, he was twice Nobel laureate. He got two Nobel prizes, once for protein sequencing, one for DNA sequencing, and he had a lot of respect, right? So Sanger Institute UK. Initially, it was Sanger Center, then now Sanger Institute. I mean, one of the famous institute in our field, working awesome, right? And they are doing this sequencing, and they are also providing us those latest research. Okay, so most of the findings are coming from their lab, right? And they're also having training programs also. So one of our joint program where one of our colleagues from Lums University, she was there, so we got connected. So that's where we explored this new tool, Docker. That's mainly for metagenomic analysis. So probably Sadia would like to cover that part also. NGS platforms perform massively parallel sequencing, during which millions of fragments of DNA from a single sample are sequenced altogether, right? So that's what is happening. There is a massively parallel sequencing in which you are millions of fragments you are taking from a single sample, right? And it's also called as high throughput sequencing, since you are processing very huge or high amount of data, so that's why it is high throughput. So more than 500,000 sequencing by synthesis operation they run in parallel using state-of-the-art sequencing methods in efficient ways. Better call it as high throughput sequencing, HTS, right? Why we call it as high throughput? Because NGS was up till this generation, and then we got that third generation also. So let's better call it as high throughput HTS, right? So what's happening over here is, since your molecule is big enough, you cannot process this whole thing all together. You break it into short fragments, right? Somebody looked at these short fragments. They're like fire of a shotgun. Okay, you hear the fire sound. That's just to wake up. I know online class participants, students, they start drowsy, they start sleeping. I cannot ask you to switch on your cameras because then it will overload our internet. So what you do is, somebody call it as shotgun method. Maybe he looked these fragments like fire of a shotgun. So how you get those fragments? You have the different methods for that. We have the instruments where you shake them, where you put the sound waves on them. It's called as sonicators, and then you break them. You set up some frequency, and that frequency cuts at a size of 100 bases, right? You change the frequency and they are cut at 150. So that depends upon the requirement from sequencers. So some of the sequencers they like at 100, some like at 150, some like at 250. That's how it goes on. Okay, so that's very important. So you take those fragments and you break it into the short fragments, right? And then those fragments are then uh, processed or sequenced by sequencers, and you put them together. Since you have multiple copies of the original DNA molecule, here we have 4 copies, and since you are doing that fragmentation at random, the other methods are chemical digestion also. You also use the restriction enzymes. So a combination of different enzymes, they also cut them at different intervals. So that's also one of the methods. But most famous is those sound waves technology, sonication or sonicators. And then you process them through instruments within those sequencers. So the instrument which Illumina contains is what we call it as flow cell. So that flow cell is something like this slide made up of glass, and that has those fine tubes or channels on it. So normally the old flow cells, they had 8 channels. So the slide is old, and I got it from a research paper. But now these flow cells are even 16, so they are double that. Okay, Solid came up with another similar idea, but these are not those closed tubes within that glass slide. These are the channels, something like open channels. The streams are channels, right? So that is where you load your material and do that sequencing. 454 came up with what we call it as pico titer plates. So they have the plates, on those plates have wells in it. So they have wells, right? Holes, deep holes in them. So there you do that. Pico titer plates, picometer size. Those wells, they have the width of picometers. Okay. DNA is in is that small. Okay. So we have pico titer plates, PTPs. Iron torrent. So iron torrent is iron semiconductor chips. So that has iron semiconductor chips, the chips which we use in your digital cameras. So you load your sample there, and this hydrogen ion, when it comes out, it it is generated and it changes the pH of the medium. So that change in pH is detected by these iron semiconductor chips, right? This is how this whole party is set up. So what you have is experimentation and extraction of DNA and RNA, and then you break them into short fragments. That step is called as library preparation. So you do experimentation and extraction of DNA or RNA. In case of RNA sequencing, you extract RNA. In case of DNA sequencing, you extract DNA. And library preps, where you break them into those short fragments and you make them ready to go into the sequencer. We also add another thing coming next called as adapters, and then you multiply those those sequences, those fragments, and you get their 10 to 20,000 copies each, because you incorporate a base and then you detect a light signal that comes out. So it's really hard to detect a light signal. You need to have then really high resolution cameras. So for that, it will increase their cost too much. So they came up with a solution rather than improving the resolution of those cameras. That was not possible. So they increase those fragments. So each fragment is then multiplied here 10,000 to 20,000 copies, and then those copies, when those nucleotides are added, they are added to all those 20,000 copies at once, and they give a very strong signal, so that can be then detected by the cameras, right? So one of the technique which these three technologies are using is called as emulsion PCR. So there is another step. Step one is experimentation. Step two is library preps. And then step three is PCR. And I marked it red because in third generation, that step has been reduced. That step has been removed. On the right is PCR on slide. So on slide they do PCR. It's called as bridge amplification. Over there some bridge-like structures are formed. Bridges are formed, so that's why it's called as bridge amplification. And then we have that ion semiconductor sequencing. 454 has pyro sequencing. The fourth layer is sequencing. Ion semiconductor 454. By the way, this 454 was the number of the project. So pyro sequencing is coming from which technology? Anybody? We just covered. Roche. The company's name was Roche. Ion Semiconductor. The company's name was Thermo Fisher Scientific. And Solid was again ABI, right? But that is again, I insist that we should drop it, but just to let you know that there was this technique. Reversible terminator technology was adopted by Illumina. So Illumina, Roche, Roche got famous as 454. Some people also call it as pyro sequencing. So 454 or Roche or pyro sequencing means the same thing, right? And then the fifth layer is data analysis. So that is when this data is generated, how would we analyze that? This course is all about data analysis, but we will cover the theory of data generation, how it is done, what is done in those machines, their comparative analysis, what's happening here, what's happening there. And obviously the amongst those NGS technologies or techniques, Illumina is the one that is, I would say, that got the more business, or that got the more famous because of their techniques and their algorithms. Okay, so this PCR on slide or bridge amplification was where they won. This emulsion PCR is bit complicated. Similarly, their reversible terminator technology, right, using 4 colors, that was another a wonderful idea. Here they are using the same color light for these technologies, right? So that's why uh, the Illumina got more success. Similarly, we see in their data analysis when they do base calling over there, they were also smart. Experimental design. So we just talk about like in our real life, means we are analyzing the data coming from different domains of different experiments. What is PCR on slide? Bridge amplification. So what happens over here is those sequences, those fragments, they go onto those slides and everything happens over there. For emulsion PCR, you need to have another instrument where you do that emulsion PCR, and then you take those amplified fragments and you load into the sequencers. Ahmed, I hope that answers your question. Right, so we'll come back to it when we go and see all these parts one by one. We talk about library preps in detail, and then we talk about those PCRs, and then the sequencing things. Welcome. So next is, in the human subject, obviously you cannot set up an experiment. Experiment is already set by God Almighty. And then in the plants, the you can set up the experiments. You can set up the experiments on animal models. You can do that on bacteria on your Petri plates. You can get the bacteria from your gut or your patient's gut or your whatever that experimental individual is, or you can get it from its body secretions, right? Or you can get it from any samples. You can get it from soils. You pick up a soil, you will have mixture of bacteria there. Okay. You pick up a water sample that has a lot of bacteria in it. So you just sequence them and then you try to find which type of bacteria you have. That's called as metagenomics. Metagenomics, mixture of genomes, right? So you have the human subject, you can have the plants, you can have the animal models, you can have the bacteria. Data analysis is data analysis, and a data analyst, we love the freedom of working with anybody. So we work with microbiologists, we work with virologists, we work with human genetics, the plant genetics, animal scientist, anybody, right? So that's the liberty, and that's amazing. If you learn data analysis, you can land anywhere. Ask for PhD positions or postdoc positions or jobs, and since you are a data analyst, you can work with multiple data sets. During my PhD, I worked with soybean, the plant, but here when I got back as a and established our group in data analysis, so we got the work from humans, we got the work from bacteria people, from even virologists. Nowadays it's gaining pace. Initially we thought maybe we don't need NGS for viruses, but after COVID everything changed, right? So anyways, so in library preps, there is one thing which we do is we take those fragments and we break them into those short fragments. And since we break them into like those fragments, and those fragments are in millions, so what we need to do is we need to sequence them and we need to amplify them. We need to do their PCR. In order to sequence, and since this sequencing method is where we are synthesizing a new strand from an old strand, we already covered that. And one thing I just forgot to tell you over there, this method is called, I'm sorry, it will much faster. No, again. Stop. Yeah, here. So that new strand is synthesized from the old strand. So that's why we call it as sequencing by synthesis, right? So NGS is sequencing by synthesis. Sanger is also sequencing by synthesis. So you synthesize a new strand, that's why it's called as sequencing by synthesis, SPS. Whereas the third generation sequencing, Oxford Nanopore and the other ones, over there you don't synthesize a new strand. So that's called as real-time sequencing. Okay, so you take the DNA as such, the big chunk, and then you get it from outside, and then you sequence it. You do it in big chunks. There, you don't need to even amplify that part. So that third layer is not there. So we are covering this second layer, library preps. We had that experiment. The experiment is set by our collaborators. Even we don't do library preps by ourselves. So people working in NGS facilities where they have sequencers, so they have the teams working with those library preps. Here in Pakistan, since our sequencing facilities are not that productive, they are not that running. We generally rely on commercial companies. We're working hard to establish our local facilities. So wherever people they gather around the sequencers, now they are looking for the money, they're looking for the funding for the projects with the people PIs to come to them and help them so that they should run their machines. So getting a sequencing facility is not a big deal. Obviously, it's a big deal. You need a lot of money, but you need way more money to run them, obviously. So we used to rely on the commercial companies. So there are the companies here that uh, provide us these facilities on commercial basis, not locally. They also work with the international companies like China or Japan or Korea or Taiwan mostly. So over there they give us the good quality data. So they do all those steps by themselves. Okay. So within that $300 for RNA-seq, my data was covered. So they did that library preps, right? So what they used to do is, since you cannot attach primers or you cannot design primers for millions of the fragments, so we can attach something which we already know. We call it as adapter. So you guys might be familiar with the universal primers or universal priming, are you? Right, so these adapters are similar concepts. So we attach a molecule of 10 20 bases which we already know, and that fragment is suppose 100 bases or 150 bases or 250 bases depending upon what is the specification of the sequencers. NGS sequencers generally go from 50 to 400 or 450. That is where we have in pyro sequencing we have longest length. In the third generation now they are going up till many kilobases, right? KBs. Okay, so that's a lot. So here we were restricted to 100 bases. That's why it's called as short read technology, right? So most successful was an economical or cheaper one was on Illumina machines, and one of them was Illumina High C. So that used to have these fragments of length 100 bases. So if the original length here of this fragment is 100, you can attach 10 bases on this side and 10 bases on that side. And they are the same all across all those millions fragments, right? So then you can design two types of primers. One for this, we call it as forward, one for this, we call it as reverse, right? So you can design those primers, and then just two types or one type of primer will be enough for them, the complementary other part, right? So these adapters, these molecules are added. So here adapters are added to this guy and this guy. These ones where the adapters are not added, they are discarded. That's called as selection or screening. So in library preps, you do fragmentation, you attach adapters, and you also do screening. And finally, you make them as single stranded. Now in single stranded, they become templates. The third step is converting them into templates, the template preparation. In library preps, you do fragmentation, you attach adapters, then you select, that's screening, and then template preparation. So these are 3 to 4 steps which we do in library preps. Right? In RNA library preps, the only difference is that RNA is quite unstable, it's degradable, so you convert that RNA to cDNA real quick. Rest of the part is the same. So one of the technologies called as paired end sequencing. So this one was called as single end sequencing. So where you can take each and every bit and pieces which you fragmented here like this, right? So here in paired end, you don't take each and every part of it. So what you do is you decide, this one is still quicker than me. So you decide this is a longer chunk, and you decide that you want to take a fragment from here. And that fragment is suppose of length of 1000 bases or 1 kilo bases. So what if I take like 100 bases on this side and 100 bases on that side, even if I remove the in between 800 bases, right? That is the 80% of that data or that DNA length. If we discard that, still we can capture that fragment. Okay, because having 100 combination of same 100 on this side and that side is impossible in another fragment, right? So what we do is we decide to take its ends. So in order to take its end, we take that piece out and we attach what we call it as adapter here on those ends. The way we attach the adapter back there, right? But that adapter is different. These two parts on right and left are complementary to each other, so it can circularize, right? Something like that. So this way we can just tie a knot here, right? They can stick with each other, and this thing becomes circular, right? And then we discard rest of the parts. We do restriction digestion, and we only take the part that is having that knot, that is having that adapter, right? That means we are actually capturing this part here and this part here. They were actually the ends of that long part, so we are capturing the ends. This thing is called as paired end sequencing or paired end technology. Okay, the rest of the party is the same. So we are not going into the further details. So this paired end method or paired end reads, we will deal that data in future, just to let you know that this how we process that in library preps. Okay, why it is important? There is very important role of this paired end sequencing in genome assembly projects, where you can have repeats in your original DNA fragment or in your original chromosomes or genomes. So you can pass that repeats uh, safe and sound. You can copy, you can recognize that there is a repeat, and you can recognize what is the length in between what is the length of the repetitive region by using this sort of technology. Suppose the repeats are there in that region that was discarded, so you get those pairs, so you can bypass those repeats. Okay, and you know the distance between those pairs, that is 800. So in 800, suppose if a repetitive region of 10 bases is there, how many expected copies are there, you can calculate that. Okay, all right. So next is the ECR part. I think I would start here, so maybe we can have 5 to 7 minutes. I can take your questions, comments, or queries, any suggestions and advice on improvement. So we did that. Today's lecture was audible, clear, understandable. You liked it or disliked it, feel free to just let us know, so that we can improve that. I cannot see your faces, your expressions, whether you're comfortable or not comfortable. In a real class, people are in front of you, and we can really capture them. Whoever misses the eye contact in my class, he or she is in trouble. I can ask them their questions, and then make sure that they are understanding the things. So please let us know how we are doing as far as this first part was concerned. So we'll resume the class from here tomorrow. Any questions, concerns, queries, comments? Most welcome. Over to you. Maybe you can write in chat box. You can write on WhatsApp also. Okay. I'll repeat what I have covered today real quick in first 10-20 minutes. That's what we do in every class. And obviously, if someone misses the class, you have the recordings also, right? And we'll share both the things once the lecture is done. These slides, and obviously this content we can share right now on your WhatsApp if it is shareable. Yeah. Any questions, comments? Are we good to go? Yes. Okay. Thank you so much. We started the recording. Stop recording, and
Excuse me, sir. I have a question.
Yes, please.
Sir, actually, I wanted to know